Nowadays, science evolve in a rate that we can't imagine. Have you thought of a human using their liver organ to test a drug? And this is owed to the Assembloid, and it helps developing effective drug miraculously. Nearly 1 in 5 individuals suffers from a psychiatric disease. What is even more striking is that the lowest success rate for finding new drugs is in psychiatry, out of all the branches of medicine, likely because until now we couldn't really access the human brain. Why do most promising drugs fail in human trials? Because we test them on animals or flat dishes, models that don't reflect how the human body actually works. But what if we could grow a miniature human brain in a dish, made from someone's actual cells? This breakthrough is called an Assembloid, and it's revolutionizing medicine. The process is surprisingly simple. Step 1, CRISPR editing. We start with human stem cells, but not just any cells. Each stem cell line carries hundreds of genetic variations, and some can acquire mutations during culture. So before any experiment, we verify genome integrity using high-resolution methods like SMP arrays, not just basic karyotyping. We also confirm the cells are mycoplasma-free and track their passage number. This rigor ensures that any differences we see later come from the disease mutation. Using CRISPR, we either introduce a disease-causing mutation or correct a genetic defect. We always include a control, editing cells back to normal, to confirm any changes are from the disease gene, not lab error. Step 2, grow specific cells. The edited stem cells are guided to become specific types, neurons for brain studies, or blood vessel cells. This takes about 2 to 3 weeks. Step 3, assemble. At exactly the right stage, we bring different organoids together. In the dish, they naturally fuse, neurons extend connections, blood vessels grow through the tissue. Step 4, validate. We confirm it works 2 ways. We check anatomically, are the right cell types present? Using special dyes, we can see if these are truly forebrain neurons or something else. We use 2 approaches to check functionally. For patch clamping, a tiny electrode touches a single neuron and listens to its electrical chatter. It's the gold standard for proving a cell is truly active. For calcium imaging, when a neuron fires, calcium rushes in. We can watch this happen in real time, thousands of cells lighting up like fireworks. For decades, drug testing relied on flawed models. Animals, but mice don't have human brains. A drug that works in mice often fails in humans. Flat dishes, cells grow in 2D, missing the complex 3D interactions of real tissue. Assembloids bridge this gap. They create a human-relevant environment where neurons connect with blood vessels. Immune cells can infiltrate. Multiple cell types communicate, just like in your brain. So when we test a drug on an Assembloid, results are far more likely to predict what happens in an actual patient. Here's a real example, Alzheimer's disease. In an Alzheimer's brain, the cortex thins, ventricles enlarge, neurons die. Using CRISPR, we create Assembloids carrying the exact gene mutations that cause inherited Alzheimer's. These mini-brains actually develop the same features, shrinking tissue, dying neurons. Now we test a new drug. We ask, does brain tissue stop shrinking? Are neuron connections preserved? Does electrical activity stay healthy? If the drug works in the Assembloid, we have much higher confidence it will work in humans. This isn't science fiction. It's happening now. So, if you had your own Assembloid made from your cells, what would you do with it?